Hi there. This video is about basic genetics. I'll talk first a bit about Mendelian genetics, and then a little bit about genes in populations and how those sort out. Then I'll talk a little bit about life cycles and ploidy levels and things like that. And then finally, I'll talk about non-Mendelian traits, things that don't really fit into what Mendel found, but nonetheless, these are also things that happen in nature and lead to some interesting evolutionary outcomes. But first, a bit about Mendel and Mendelian inheritance. Starting in the 1850s, Gregor Mendel, who was a friar and an abbot in Europe, started his work to try to breed garden peas to make some improvements. They grew a lot of their food at the abbot. So his function was to work with these peas. So he started his experiments, and eventually his results were published in 1865 and 1866 in a paper entitled Experiments in Plant Hybridization, and there's the German title. It was later translated into English, but from 1865 to 1900, it was cited only 3 times. So he died in 1884, thinking that no one had even noticed what he had done. But in 1900, 3 researchers independently rediscovered his work, De Vries, Correns, and Chermak. And so, modern genetics had its start at that point. Mendel described these morphological characters as Merkmal, which is the German for characters or features. And so that's all he knew, that these were characters that he could distinguish among the plants. So he knew nothing of genes. He didn't know what alleles were, linkage groups, chromosomes, recombinations, haploids, diploids, or DNA. These all were much, much later discovered and described. So some of the basic Mendelian characters were flower color. So he had purple flowers and he had white flowers. He had yellow seeds, green seeds, things like that. He did not know what the genotypes were. There were no words for genotype, and he didn't even know what a gene or a genotype was. So he narrowed it down to some characters that were predictable from one generation to the other. In his paper, if you ever want to read that, he also described some other characters that were not as stable and could not be measured by him. So he carefully recorded the colors and each of the characters that he could measure from one generation to another as he both cross-pollinated and then allowed the plants also to self-pollinate, which most legumes will do. It's very common. So the characters he saw could be bred so that they would breed true. So he started breeding just purple colored plants with purple colored flowers. And if he did this one cross after another from one generation to the next to the next, then those characters, in this case the purple color, would breed true. All of the progeny would have purple flowers. He also discovered that if he had the occasional plant that had white flowers, if he took the seeds from those white flowered plants and bred those for generations, that they also bred true. So for several generations in a row, you could get white flowers and only white flowers on those plants. When he took pollen from purple flowered plants that had bred true and fertilized a white flowered plant with that, or vice versa, took pollen from the white flowered plant and put it on the purple flowered plant to fertilize it, In all cases, he got, in the first generation, got all purple flowered plants. However, if he allowed those plants to self-pollinate, then the F2 generation always had some purple flowered and some white flowered progeny in the ratio of 3 to 1, 3 purple to 1 white. And so he started calling these the dominant traits for the purple in this case, and the recessive traits. That is, it was hidden in that first generation. So there's a recessive trait that was the white flowered plant in this case. So eventually in the 1900s, then, it was found that these plants and many eukaryotes were diploid, and they carried 2 alleles for each gene, sometimes more than that, but just in the simplest case, so that the plants that bred true and had purple flowers had 2 dominant alleles, BB, and the white flowered plants in this case had 2 recessive alleles, small b, small b. When those were crossed, then they formed heterozygotes. When those heterozygotes were allowed to self-pollinate, then those alleles separated because the gametes were haploid, and then when those were crossed, then you got a ratio of 3 purple flowers to 1 white flower, and they were actually in the ratio of 1 to 2 to 1, the homozygous dominant, the 2 heterozygotes, and the homozygote that had the recessive alleles. So Mendel was able to measure and carefully record 7 characters that had these so-called Mendelian traits that we call them now. So flower color was one, where the dominant condition was purple, the recessive was white. Flower position, where axial was dominant, that is, borne on the side of the stem. Terminal at the end of the stem was recessive. Plant height, tall versus short. Pod color, green versus yellow. Pod shape, inflated or constricted. Seed color, yellow was dominant, green was recessive. And then seed shape, round versus wrinkled. When he measured 2 traits at a time, in this case, seed color, yellow versus green, and seed shape, smooth versus wrinkled, he always came out with a 9 to 3 to 3 to 1 ratio. That is 9 yellow smooth, 3 yellow wrinkled, 3 green smooth, and 1 green wrinkled. Ultimately, now we know, as the alleles separate in those from the crosses of the heterozygote, we can tell how those alleles come together in the diploid, as shown in this Punnett square. So now if we go back to this simple diagram of the just the flower color, or you could use any one, you can then build ratios on this and start testing the allele frequencies both in the F1 and the F2 generations. So here's what I mean. On the left-hand side, you can look at the female gametes, male gametes, how they come together in the progeny. So you have the 2 dominant alleles, the heterozygous, coming together, and then the 2 recessive alleles, and they're in a ratio of 0.25 to 0.25 to 0.25 to 0.25, or 0.25 to 0.5 for the heterozygotes, to 0.25 for the recessive homozygote. But that's when the ratios of each of the alleles, the dominant allele and the recessive allele, are both in a 50% or 0.5 to 0.5 ratio. For haploid organisms, the allele frequencies are dependent on the size of the immigrant population or the extant population and the relative fitness of the 2 alleles. For example, if allele A1 has a frequency of P and allele 2 has a frequency of Q, then with relative fitnesses of W1 and W2, then the eventual frequency of A1 and A2 will be equal to the proportional sizes of each population and the relative fitness of each allele. The alleles can be in the same population also, so it doesn't have to be an immigrant population in this case. For diploid organisms, the possible genotypes would be A1A1, A1A2, and A2A2, with relative frequencies of P squared, 2PQ, and Q squared, respectively. Hardy-Weinberg then came up with an equation that addressed this, and it was mainly looking at whether these two alleles are in equilibrium or not. And it's mainly to look, if they're not in equilibrium, what else is going on. So essentially they said the frequency of the A2 allele, that is Q, equals 1 minus P, which is the frequency of the A1 allele. So P then would equal P squared plus 0.05 of PQ. So in words, this means that the frequency of A1 is equal to the frequency of A1 in the A1A1 homozygotes plus the A1 alleles in the heterozygote. If you include the fitness in the equation, then you have to include the fitness of the homozygote for the A1 allele, the fitness for the A1 in the heterozygote, and the converse for the A2 allele having the W22 and W12 frequencies. So then you come out with the equation below, which shows you what the new proportion of Q is, Q prime, in the population. So normally if there's equilibrium, then Q, either Q or Q prime, should be at 0.5, and P should also be at 0.5, meaning that there's no selection. If those don't equal 0.5, if they're like 0.75 versus 0.25, then there's selection for one allele over the other of some sort. This is a graphical representation of the same thing, so you can look at the genotype frequencies there. If you have the homozygous dominant allele, which has a proportion of P squared, you can see it goes from 0 to 1, and the same for the homozygous for the recessive allele, or Q squared. And then in the middle is the proportion of the AA, the heterozygote, which is 2PQ for the frequency. And so at equilibrium, where each is 0.5, then you can see that the proportion of the homozygotes is 0.25, and the proportion of the heterozygote in that population is 0.5. So that's at equilibrium, but if you're not at equilibrium, meaning there's some sort of positive or negative selection going on, then if the dominant allele is selected for positively, you would move further to the left on the graph, and if the recessive allele was under positive selection, then you would go further to the right on this graph. If you look at gene frequencies in populations, and they can vary from one environment to the other. So some genes in one environment are favored, but in a different environment, the other gene is favored. In the first case on the left, if there's no selection, then the gene frequencies can change independent of one another. However, in environment 1, if there's selection against A, then you'll see it in lower proportions than the B gene. In In environment 2, it might be the opposite, where gene B is selected against. So you'll see an increase in the gene A, or gene A, or it can be an allele also. On the other hand, if you have selection in favor of A, it can increase in environment 1, where the converse is true in environment 2. If there is linkage between the two genes, that is, A and B are linked, then if you have selection in favor of A, then both will increase in frequency. And in environment 2, if you have selection in favor of B, then both will also increase in frequency. Conversely, if there's selection against A, then both A and B, if they're linked, will decrease in frequency. And on the other hand, in environment 2, if there's selection against B, both A and B, if they're linked, will decrease in frequency in that environment. If you follow particular alleles in a population, in the case of an advantageous allele, often they either disappear right away, or if the population is small or they're very advantageous, then there's rapid fixation in the population, and they will stay there in that population, remain for quite some time. Neutral alleles can go extinct. They can sort of increase, decrease, sort of randomly in the population, and they can either go extinct or some actually can become fixed. And neutral alleles are by far more common than things that are either advantageous or disadvantageous. There are several possibilities for allele proportions within a population. Some alleles go to extinction rapidly, some go to fixation rapidly, but more generally, there are some that are stable. The most common are those that are erratic, that is, they will go up and down, and this is true of most neutral alleles in the population. So here's a theoretical example where you start with two alleles, A1 and A2, and they vary in proportion within the population. Then a third allele arises due to a mutation or gene flow of some sort, and that goes to rapid extinction, followed by the rise of a fourth allele and the extinction of the A2 allele, and then finally a fifth allele pops up and briefly and then goes extinct, and eventually the A4 allele goes extinct, and all you're left with in the population at the end is the A1 goes to fixation and is essentially 100% in that population. Population size has a large effect on the proportions and changes in those allele proportions. So in a small population at the top, you can have alleles going to fixation rapidly within the population. You can also have alleles going to extinction rapidly, and all the others are pretty much erratic within the population. For a moderate-sized population, this narrows around the mean of 0.5. And then for very large populations, then these tend to concentrate around the mean of 0.5. And so for a large population, changes in allele proportions aren't very likely, but for small populations, the allele proportions can change very rapidly and very drastically. And this is why, in some cases, small populations of certain organisms can be in danger of rapid extinction as well. Here is a conceptual diagram of this. So if you start with a population where you have different proportions of different alleles, different genes, and that population shrinks, then you'll have a smaller number of alleles or genes to choose from. If that shrinks even further, such as over here, then you have what's called a bottleneck, where you have very, very low gene diversity and allele diversity within that group. Once those expand again, you still have low diversity because you started with a population that had a low diversity, whereas the population in the middle there, when it grows, it will have a greater diversity and so a greater chance of surviving in different environments or different conditions. Populations seldom are totally isolated, so over time there's immigration, which is called gene flow. So you have new alleles, new genes entering a population. Depending on the size of the immigration population as well as the original population, the results can vary from having no gene flow, even though a few individuals enter that population, to having an allele or a gene completely take over within that population. And that would usually only happen when it's a very advantageous allele or gene. Here is an example, and so in the middle you have populations 1, 2, and 3, which correspond to having one allele, two alleles, or three alleles. And those then blend with one another in different proportions. So a large dark arrow means there's a large contribution from that population, and a small arrow indicates a small contribution. So when they mix, you can see the different amounts of each of those alleles that wind up in those particular populations. And this, of course, is the case where there's no selection. So it's just essentially blending of those alleles within those various populations. If there were some selection, then you would see those proportions change, either positively for positive selection or negatively for negative selection for those particular alleles. Here is just one example, that is wind transfer of pollen and seeds in plants. So in some cases, you'll have gene flow, that is seeds blown for a short distance and then plants come up from there. And so they're contributing to the genetic makeup of the population through that movement. Pollen moves much further, so you can have pollen moving to up to several thousand kilometers sometimes, and then that will affect the population, the allele proportions in the population where they wind up, and then again you'll get either neutral or positive or negative selection for the alleles that are entering those populations. Taking all of those criteria and mechanisms into account, then you have mutation, selection, gene flow, gene drift, and other factors. So you can start with allele A, you have mutation and selection. Eventually at a different time you'll have other alleles, A, B, C, etc. And then you're dealing with the proportion of allele A plus the proportion of allele B, etc., all the way down until the next generation, when you have more mutation, selection, gene flow, etc., then you'll have a different proportion of those alleles and more mutation selection after a certain time. And so eventually you wind up with a fairly complex arrangement of alleles and movement of those alleles through the different populations in the different areas and different environments. So I've shown this figure before in some of the previous presentations, but I wanted to cover it one more time, because now I'm going to talk about differences between populations of unicellular organisms and then populations of multicellular organisms. So this is all the light areas, remember, are those are the proportions of unicellular organisms in each one of those groups, and then the dark areas are the proportion that you have of multicellular organisms. in each case. So the genetics, the movement of alleles, the selection varies quite a bit among all of these different taxa. So one of the reasons for a lot of this variation is that there are many, many different life histories for organisms, ranging from large organisms to very small, including unicellular organisms, organisms that produce only a few offspring during their lifetime, and others that produce hundreds to thousands of offspring during their lifetime. The sizes of the offspring can be large or small, and going down the line. So you can have essentially different combinations of each one of these, creating a different form of organism, a different lifestyle, where they live, how long they live, how large they are, etc. Even when looking at a fairly limited sampling of organisms such as animals, plants, and fungi, you find quite a bit of variation in their life history. So in animals, much of the time, for at least most animals, the vast majority, is spent as a diploid organism, or the sporophyte. And a very small amount of time is spent as a gametophyte, that is, a haploid organism. In fact, in humans, the gametophyte generation is only a single cell, unicellular, the sperm and the egg. For higher plants, most time is spent in the sporophyte. So the trees that you see, that's the sporophyte generation. And most of the cells there, except in the flowers and the cones, are diploid. And the gametophyte is a smaller part of the life cycle, although it's usually multicellular. So both gymnosperms, angiosperms, and other plants have a gametophyte generation, a haploid generation, that eventually form the sperm and the egg, but they actually have a haploid multicellular generation. In lower plants, actually, the gametophytes take up most of the life cycle. So there's a long and prolonged gametophyte generation and a short sporophyte generation. In fungi, at least in the Dicaria, the Basidiomycetes and Ascomycetes, there's not only a diploid generation, which is usually short, there's an extended haploid generation, usually hyphae, or yeast cells. But then there's a dikaryotic stage where each cell has two nuclei, one from each parent, and those divide so that there are two nuclei in each cell in the hyphae. They also have multinucleate stages where the dikaryotic cells and the haploid cells often multiply their nuclei without cell division, and so there are multinucleate cells as well. So quite a few different ways of distributing the genes and the alleles within each one of those populations and multicellular organisms. Here are a few examples of those in plants. You have the 2N version, the diploid plant, which is a sporophyte. And then you have the flowers. This is in an angiosperm in the female parts of the flower. Then a carpel forms, including the ovaries and ovules. Those become multicellular, and in many higher plants, you have mitotic divisions of those haploid cells, becoming 8 cells. So that's a haploid generation. The gametophyte generation is about 8 cells, although other plants differ in those numbers. Also in the anther, you have haploid mother cells that then go through a mitotic division of the haploid, eventually forming 2 sperm nuclei that move down the pollen tube, and another cell also, or another nucleus. And one of those sperm cells then fertilizes the egg cell in the female ovary. and the other sperm nucleus fuses with the polar nuclei, initially becoming the endosperm, which is triploid. So again, different shuffling around of different chromosomes and different alleles in those fertilization, gametophytic, sporophytic, and fertilization events. In animals, including humans, of course, the gametophyte generation is only one cell, that is, a sperm and an egg. And so there is no extensive gametophytic generation or tissue formed in those organisms. And most of the cells in the organism are the sporophyte, the diploid organism. There are many variations on the major themes in animals. This is an arthropod, an aphid life cycle. So in the spring, all of the individuals born are female. There are no males. And so during the summer, when the feeding on the plants occurs, the female, by parthenogenesis, gives birth to clones of herself, small little aphids that then grow into females and do the same thing. And so you have clones and clones based on just a few females. Later in the summer, she sprouts wings. She also gives birth to a few males, which are missing one of the chromosomes, and that's what makes them males. And then there's mating between the males and the females. The female, in this case, does lay eggs, and these eggs are all female eggs because they have both of the X chromosomes. And then the cycle repeats during the next year. For fungi, and this is an ascomycete, the haploid hyphae are formed from haploid spores. They're of two or more mating types. Once they find an opposite mating type or a compatible mating type, they anastomose, and so the nuclei proceed into to mix into the cytoplasm of the cell that the cells that have come together. Some of those cells become multinucleate, and they bud off the dikaryotic cells. And the dikaryotes have one nucleus from one of the parents, one of the mating types, and one nucleus from the other mating type. Eventually a crozier hook is formed, and that the two nuclei in that one cell fuse to form a diploid, and it's a short-lived diploid because it immediately undergoes meiosis, forming 4 haploid cells, and those cells then divide mitotically, forming 8 of these haploid cells, and then each one of those eventually comes off, is shed, and is then germinated again, and you repeat the cycle. Many fungi have both a hyphal life cycle and a yeast-like life cycle. Some form hyphae and some form pseudohyphae. This is Saccharomyces cerevisiae. So again, you can start with haploid spores. Those then germinate into unicellular yeast cells, and then you get budding occurring over and over and over again, going from a haploid to more haploid cells. These can then mate with the opposite mating type to form a diploid. The diploid can actually undergo mitosis and mitotic divisions over and over, so you can have both a haploid population and you can have a diploid population. These can then do one of several things. One is to form pseudohyphae, one is to die, one is to essentially go into a resting state, and then one is to go into meiosis, followed by shedding of those spores, and then repeating that cycle. So again, if you look at the alleles that are the alleles and genes in each one of these different stages of the life cycle, whether it's haploid, diploid, tetraploid, triploid, or whatever, there's a different combination of those genes and expression of those genes along the way. So there are many checkpoints as far as which way to turn, and there are many alleles that will cause different individuals and organisms to go one way or the other on some of these pathways. Plants can go through vegetative reproduction as well as sexual reproduction, and many plants only do vegetative reproduction. A percentage of plants are only vegetative, and they never do sexual reproduction. You might think that's a dead end, but there are also some animals, some nematodes, that are never go through sexual reproduction. They have hermaphrodites that just essentially produce clones of themselves. Many fungi also have an extended haploid stage, and they've never been observed to go through a what's called a perfect stage or the reproductive stage. And so many organisms for millions and millions of years, they may have once been sexual organisms, but now they're asexual organisms. So there are some plants that reproduce only through underground rhizomes. There are some, like Kalanchoe, which makes little tiny plantlets on its leaves, and those drop down and grow. There are some that form runners, such as strawberries, that make new plantlets. Of course, potato, it can undergo sexual reproduction, but it can also go through vegetative reproduction, as can many species that form bulbs and bulblets. And then most plants can also go through sexual reproduction, where they form flowers or cones or whatever the reproductive structure happens to be. However, in higher plants, there are a number, 30 or 40%, that can also go through extensive vegetative reproduction for years or decades or centuries before they become reproductive. In genetics classes, you've probably learned about Mendelian characters, and maybe you've learned about non-Mendelian characters also. So Mendel studied these characters that were predictable and that he could actually make some sense out of. But there are many characters that are non-Mendelian for a variety of reasons. Here's the first example, and it has to do with a coloration pattern in animals called agouti, and you probably have seen this in many animals. But it's where the color of the hair varies from one part of the hair to the other, and in most cases, a pure agouti is kind of a yellowish color, but then it can be mottled with different types of coloration pattern, and this has helped with camouflage to avoid predators. Now, if you cross heterozygotes at two different loci, the A locus and the C locus, instead of getting a 9:3 to 3:1 ratio, which you would expect from a typical Mendelian pattern, what you get is a 9:3:4 segregation in the morphologies of the color pattern in those mice. This occurs when the expression of one gene and one set of alleles is dependent on another gene with another set of alleles for its expression. So in this case, the dominant A, is responsible for the agouti coloration, which is sort of a sputter pattern of turning on the pigmentation gene, whereas the double A recessive allele causes a black color, which means the coloration is turned on fully in all of those cells. On the other hand, the C locus turns on that A locus. So if you have either a CC dominant, the large C, or you have a large C, little c, then the coloration is turned on. Whereas if you have a double C recessive mutant, coloration isn't turned on at all. So then you can see you have the 9 agouti, the 3 black, because the recessive A alleles, and the 4 white, because of the recessive C alleles. Here is another example, in this case for flower color. So again, both genes, the dominant allele is needed for both genes, both the color gene C and the purple color P gene. So as long as you have at least one dominant allele of each of the genes, the C and the P, then you get a purple color. But if you have a double recessive set of alleles for either the C locus or the P locus, then no color is formed, so you get a white flower. So in this case, you get a 9 purple to 7 white ratio in the progeny. The reason for this is that each one of those genes, the C locus and the P locus, encode an enzyme. So they are within a pathway that forms the purple pigment. So if you do not form the C enzyme, then the pathway is stopped. And even if you have the C enzyme that's produced, if the P enzyme is not there, then the purple pigment is also not produced. Just the precursor is there ready to go. So here's another example of the ABO blood type groups, which essentially are Mendelian in a way, but then are a bit confusing also. So on the cell surface of the erythrocytes, there's a protein that sticks out that has a set of sugar groups attached to it by specific enzymes. So there's the AB, and the O type. So the genotype of A is either the AA or the AI, and the I means that there's not a gene there to add any sugar group to that. The BB is there, or the BI, which is again hemizygous, and it has the protein to make to put the glucose on there, but not another allele on the other chromosome. The AB is a heterozygote, and then the O has no allele at those two positions. Individuals that are homozygous for the A locus, or they have an A locus, an A allele, and on the other chromosome there's nothing, they're hemizygous, they make antibodies to B. Those that are homozygous for B or hemizygous for B make anti-A. Those that are heterozygous for A, B, make no antibody to A or B. And those that are O are homozygous for no allele at that site, and they make both anti-A and anti-B. Because of this, then the AB individuals are universal, not donors, but acceptors of blood transfusions. They can accept from anyone. And the type O are universal donors, but they're very restricted on what type of blood they can accept. They can only accept also type O. Another example of hemizygosity occurs in the sex chromosomes of Drosophila melanogaster. And in this case, the X chromosome in a male carries an allele, whereas the Y chromosome does not have a gene in that position, so it's hemizygous. So if you cross an eosine-colored eye male with a red-eyed female who is heterozygous for the red locus, that is, the locus, the allele on one chromosome is R, the red color, and the allele on the other chromosome is for the white eyes. Then you get a two, in the progeny you have two red-eyed, one eosine and one white-eye. So the red-eye are the ones where the female donated the red chromosome, and the eosine is on the other chromosome of one, and on the Y chromosome, of course, there's no allele. Whereas the eosine individual is a female carrying the eosine allele on one chromosome and the white allele on the other chromosome. And the white-eyed individual has a white allele on the X chromosome and nothing on the Y chromosome. And we already discussed in previous lectures the non-Mendelian characters in broadbean as well as in flax, peas, other higher plants, where you can have one chromosome carrying a certain number of genes and the other chromosome carrying a larger or smaller number of genes, usually caused by unequal crossing over, which is shown here on the Vishafaba chromosomes at the secondary constriction. You have all of the ribosomal genes, and this is common in higher plants. But you can get crossing over because of the repetitive nature of those genes. They can cross over in many, many places along the chromosome. And so after a crossover, one of the chromosomes has lost a number of those genes, sometimes in the thousands of genes at a time. And the other one has gained those genes onto it. So instead of having 2,500 genes average, you get a crossover, and one of the chromosomes winds up with 1,500 copies, and the other winds up with 3,500 copies. And you can get larger or smaller crossing over events on those chromosomes. So in Vishafaba, anyway, you can have as much as a 12-fold change in the number of ribosomal genes within an individual plant, and up to a 95-fold change within a single population. We also looked at histone genes, and you can get up to a 7-fold change in the number of histone gene copies. And so a lot of DNA is being moved around from chromosome to chromosome in these cases. Another example, which I talked about before, is in flax, Linum usitatissimum, where there are 3 genotrophs: a small genotroph, which has about 136 million base pairs of DNA in its haploid genome, a plastic genotroph with about 150 million base pairs, and a large genotroph with about 158 million base pairs of DNA in its genome. Also, the relative weights or masses of these plants are: the small is about a relative number of 1, plastic 2, and large 6. Here's what they look like. Here's a large genotroph and a small genotroph, and the relative masses of each one of those is about a 6-to-1 ratio. As I discussed in a previous lecture, with the plastic genotroph, if you grow it in a high ammonium fertilizer, then it will change along the length of that plant. And so the seeds that you get off that will all produce large genotroph plants with more DNA in them. On the other hand, if you take the plastic genotroph and grow it under somewhat stressful conditions, it will revert into the small genotroph, that is, all of the progeny from that plant. If you sow those seeds, will be of the small type, which will have lost some DNA and mass of the plant as well. And these will breed true for several generations, although eventually, if you grow them under unstressful or non-stressful conditions, the large genotroph will revert back to the plastic type, and the small genotroph will also revert back to the plastic type. So these are definitely non-Mendelian traits that are happening within these plants. I don't know what Mendel would have made out of them, but large pieces of DNA are being added to the genome or subtracted from the genome based on the environmental conditions. They've been traced down to activity in transposable elements, certain transposable elements that are moving pieces of DNA around in those nuclei on the chromosomes, changing expression of certain genes either by activating them or deactivating them. Another non-Mendelian character is genome duplication, which we know now has occurred many, many times throughout evolution. And this forms polyploid cells. Most of the time when this happens, it's probably not advantageous, and it might actually lead to death of that particular individual. But in many cases, there have been polyploidization events that then has led to an expansion of the number of genes in those organisms, of course, and fairly rapid evolutionary changes in those organisms. So, for example, tracing down the Hox genes, the homeotic genes in animals, the ancestral chordate had one set of these Hox genes, 14 different members, going from that red one, which is number 1, to the gray one, which is number 14. And then there was a whole genome duplication, which also duplicated these genes, followed by mutation and evolution of those genes, and then you got the jawless fish, such as lampreys and eels and things like that. There was a second duplication that led to cartilaginous fish, so now there are 4 copies, and we have those 4 copies too, although mutation and evolution have caused some of those copies to change from what are in cartilaginous fish, which are sharks. So we're along that same line. Diverging from that were the bony fish, and they had another duplication, so they now had 8 copies of those Hox genes, and then further mutation, diversification into all the fish that we see today. So you can trace these Hox genes back further, so going backwards from the mouse and the fish, and then the cartilaginous fish, the sharks, etc. You can go back to some of these chordate ancestors, including lancelets, sea squirts, and tunicates. And then off on different lines, you can also see these Hox genes that have been inherited from the same set of Hox genes that we have, but then they've diverged in other ways to form many, many different body plans in those organisms. This is just a model of how some of those polyploidization events can occur, and then mutation after that. And so you can have the polyploidization forming duplications of the chromosome sets, but then you can have chromosomes breaking off, recombining, changing in length by quite a bit, and even exchanging genes among them, and then some genes can mutate. So in the end, even though you start with a set that is common between 2 groups of organisms, they can be quite different after a while, even within things like the Canis species, the dog species, and wolves. There are these chromosome changes that have occurred within them, and then, as you've seen from the cartilaginous fish to the bony fish, there have been a duplication and then diversification of the Hox genes from there. So again, as I've shown you, as the Hox genes change, as you got those duplications, mutations, diversifications, this causes large changes in those organisms, especially in this case, just showing some of the morphological changes in the appendages, the fins moving to more of like flipper fins to lobe fins, and then finally to things that are feet and hands and eventually wings. And the Hox genes aren't the only things that have changed. So this is just illustrating one set of genes that have changed during the polyploidization events. And these are really nothing that Mendel could have anticipated. So although we start with Mendel as some very simple, predictable events and characters, we end here with a lot of different things that can happen that have been cataloged in the years since, especially since molecular biology and molecular evolution have been ongoing in the past 50 years. Okay, so that's just a basic run-through of genetics all the way from Mendel up through the advent of molecular techniques and the finding of many non-Mendelian characters as well. This will lead us into talking about gene duplication, multi-gene families, and then eventually multicellularity in organisms. Okay, thank you very much. See you later.